Welcome everyone to this video over biology basics. This lab is going to cover a number of topics that are going to help make your learning anatomy and physiology more understandable as we move forward in the semester. We're going to be talking about how molecules move across the cell membrane, the ways in which they do that, like diffusion and osmosis. We're going to look at how solubility can affect molecules. We'll take a look at pH, and then we'll look at the cell cycle and mitosis and finish it up with a conversation about field of view. So this is a very eclectic lab with lots of different activities for you to complete. So this is a diagram you've already seen before, the atom to atom hierarchy that I've described before. And this lab is going to be discussing how is it that molecules and macromolecules are able to interact in cells. And that is really the focus of this lab. How is it that water and molecules are able to interact with cells? Clearly that's an important topic as you think of the trillions of cells in your body. Each of those cells has its own metabolism. Each of those cells must get its nutrients, must communicate with each other through chemical and hormonal signals, and then finally, cells must get rid of their waste products. And so there must be a very organized way by which molecules move into and out of cells, and that will be the topic of many of these slides in this presentation. First off, let's just think about the cell membrane or the plasma membrane. And every one of your cells is surrounded by a phospholipid bilayer. You'll hear that term a lot. It is a very complex arrangement of lipids and proteins that allow the cell membrane to act as both a barrier and a gateway, meaning that it acts as a barrier. It stops molecules from going in or out of the cell, but at the same time, the cell membrane is equipped with multiple mechanisms by which molecules can get into or out of the cell. So the cell membrane is both a barrier and a gateway. Two terms that are part of this conversation. One is selectively permeable. So selectively permeable means exactly what it sounds like. It means that there is a selection process. There's a selection process that allows certain molecules to cross into or out of the cell membrane. This could be based upon the size of the molecule or on the charge of the molecule. This could be based upon the type of molecule. There are many, many different transport mechanisms that allow molecules to move into or out of cells. So not only is the cell membrane said to be selectively permeable, but the membrane is also said to be semi-permeable. These words are very similar, but there's a slight caveat. Semi-permeable means that some molecules are not selected to come in or out of the molecule, but they just can. So some very small molecules, like water, gas diffusion in and out of cells, like oxygen and CO2, some very small molecules are able to get across the membrane even without a, quote, selective process. So we'll see that as we move into this discussion. So there are two main ways by which molecules can move in any direction, but we're going to be talking about both how molecules just move in air or move in water, but we'll also be discussing how molecules move across the cell membrane. But the two main types of transport categories, there's passive and there is active transport. So passive transport are going to be mechanisms by which molecules can move, but they do not require energy of any sort. Most oftentimes in these conversations, this means that molecules are moving without the help of ATP, without the use of ATP or cellular energy. The other main category will be active transport. We won't be spending as much time with active transport this semester. However, active transport does require the use of ATP usually, or some sort of cellular energy for that movement to occur. So let's look at some examples of passive transport. The first is filtration. This is no different than going to the beach with a bucket of sand and a pail. We've all sort of filtered out maybe beach water and filtered out the sand. This filtration always requires a membrane. So that could be a cell membrane. And there's always some pressure involved. So there's always something allowing for the water to pass through the membrane, and that either does or doesn't allow other molecules to pass with it. So from a biological standpoint, this is how your kidneys are going to work. When we get to the urinary system later in the semester, you'll better understand this. But as your heart transports the blood through your body into the kidneys, there's a certain amount of pressure that blood is pushed and is entering into the kidneys. That push, that pressure is that hydrostatic pressure, right? Hydrostatic pressure. And only very small molecules are able to pass through that filtering process and become part of the urine, part of what you'll excrete. Other examples might include just like a tea bag or a coffee filter, whereby there's a certain amount of volume, of pressure, gravity is pulling that water down through the membrane, and then some molecules are able to pass through those very small openings in the filter. What we'll spend most of our time thinking about will be diffusion. There are a couple of different types of diffusion. The first is simple diffusion. And all this is when particles spread spontaneously, right? Spontaneously without energy. When molecules spread spontaneously from regions of where there's a higher concentration to regions where there is a lower concentration. So here you can see in the diagram, right? If you were to add some food coloring into a beaker, at first the dye molecules would be concentrated in a small area, but they would over time spread, right? Over time they would spread to the point where you would see no longer a concentrated drop of dye, but you would now see that the entire beaker has that dye molecule all around. So there's a uniform color if we're thinking about dye molecules. Well, even if it's not a dye molecule, this would be true of any molecule that you added into water. As long as that molecule is able to mix in the water, you would expect after a short amount of time that molecule would be completely dispersed and equally distributed in that water. And that is a very great example of just simple diffusion. Another one would be if you open up a bottle of cologne or perfume, right? The room in which you open it, you're going to smell that perfume rather quickly. You're not going to smell it on the other side of the room, but given enough time, right, that perfume will diffuse and move through the air from the area of higher concentration to areas of lower concentration until the entire room is saturated, or at least the entire room is covered with a uniform concentration of that scent. Now, the difference between the concentrations of point A, so let's say point A, if you're talking about the perfume, where you opened up that bottle of perfume, the concentration of that odor molecule in the air from where you opened it to maybe the back corner of the room, the difference in those concentrations from point A to point B is referred to as the concentration gradient. The concentration gradient. And pretty easy to imagine, right? We've experienced this sort of idea before. So you get this concentration gradient. I'm just going to put CG here for now. And when I ask the question, which direction will the substances diffuse? right? They're going to go from an area of high concentration to an area of low concentration. Let's take a look at a petri dish. And in this petri dish, there's some water, and a crystal of some sort has been placed in the center. You could imagine this being a drop also of food coloring. And we know what's going to happen, right? Initially, there's just going to be a glob of that dye in the very center, and then over time, we're going to see that molecule move toward the areas of lower concentration until an hour from now, or maybe only 5 minutes from now, you would come back and you would see that that dye had distributed evenly throughout the entire petri dish. What I want to describe to you here are these terms. When the molecule moves from the area of higher concentration to the area of lower concentration, we're going to use this phrase, that the molecule is moving down its gradient. Now, when molecules move down their gradient, that is most oftentimes spontaneous. Okay? It is passive. It doesn't require energy. But there are situations where molecules will move up their gradient. Now, that is not something we experience in nature very often. You would not expect to have a petri dish full of dye, and the next day come back and see all of that dye had concentrated into the center, right? That would require that the molecules moved up their gradient. Now, the only way that's going to happen is if something comes along and pushes or forces, or in this case, used energy for that to occur. So let me assure you that whenever things are going to move up their gradient, it means that things are going from a lower concentration to a higher concentration. And whenever that happens, it would require energy and would be an example of active transport. So moving down the gradient would be passive transport. If you see something moving up its gradient, you can be confident that it's requiring energy and is some sort of active transport process. So in this diagram, we're adding one extra feature. What you're seeing here is a beaker. The beaker is separated by a permeable membrane. All that means is that it's a separator. There are small openings in this membrane. Remember, membrane is just something that separates, right? We've learned in lab about serous membranes. We know that serous membranes are thin layers that separate compartments one from another within the thoracic or abdominal cavity. So a membrane here is permeable, meaning that molecules can move across that membrane. Now, how permeable a membrane is would depend upon the size of the openings. But all we know here is that this is a permeable membrane. It tells us in the key that the red spheres are solute molecules. In other words, something's been dissolved. That's the solute. That's shown in red. And the blue spheres in this diagram represent water molecules. So at some point in time, let's say this is time 0. At time 0, the solute molecules are added over here on the A side. And at time 0, there are none of these molecules over here on the B side. We see the B side is purely water. Now what happens over time? As long as the molecule, the red molecule, is small enough to move through this membrane, we know that some of these red solute molecules are going to make their way over to the other side. But don't forget, not only are these solute molecules going to move from the A side to the B side, but there's also going to be the movement of water from the B side back to the A side, right? If it's a permeable molecule, then the molecules can move. And at some point in the future, who knows, this could be an hour, this could be 5 minutes, it could be the next day, but at some point you will find that the molecules have moved such that the concentration is the same on both sides. So now we have an equal concentration on A side and the B side. But what I want you to think about is that even when you have an equal concentration of molecules on each side of that membrane, there's still going to be some movement of molecules back and forth, right? That membrane is still permeable, and so molecules will move from one area to another. Now, the rate that those molecules move back and forth may not be that great, but still there would be some movement of molecules. Again, this is still passive. This is still a process by which energy is not required. So what would cause molecules to move more quickly from one side to the next? What would cause a perfume to travel from one corner of the room to the other side of the room more quickly? So let's think about some things that can affect the rates of diffusion. And a couple of them are, I think, very, very obvious, and a couple more we'll discuss. So number one, temperature. Temperature. On a warm day, molecules are going to move more quickly than on a cold day. And that's true in the air, that's true in water, molecules in water. If you were to take a drop of food coloring and you had two different beakers, one beaker was cold water and one beaker was hot water, and you put a single drop of food coloring into each beaker, right? So we had a cold water beaker and we had a hot water beaker, and we add a drop of food coloring into each. In which of those would you expect the dye to move more quickly? I think we appreciate that the dye will move more quickly in the heated water. And the reason is those water molecules, when they're warm, are moving. So think about boiling water, right? When water boils, you see it physically move. So with heat comes movement of molecules. So on a hot day, the air molecules are moving more quickly. On a cold day, the molecules are moving more slowly. So temperature does have an effect on diffusion. Another one, molecular weight. A very large molecule that is, just a larger molecule would have a larger molecular weight. It's just much easier for that mouse to move through the trees than it is for the large elephant. So just think about the size of the molecule, and smaller molecules will typically move more quickly. Large molecules will move more slowly. Steepness of the concentration gradient. In other words, how concentrated is that perfume, or how concentrated is that drop of dye? So the more concentrated the source, the more quickly it will diffuse through the environment. So again, I've got two different word stories or two word pictures going here. I've got the idea of molecules moving through water. I've also got this idea of maybe a perfume moving through air, but either one, right? Whichever one you feel more comfortable imagining, you've got molecules with a higher concentration gradient, and they're going to move more quickly. Now, the next two details, I want you now to imagine that there's molecules moving across a membrane. Or let's think about filter paper just for a moment. So if you have a piece of filter paper and you have a bucket of water, right? You have a bucket of water. It's got sand. Like, let's say we're at the beach. We've got two equal buckets of sand water at the beach. And you have two filters. You have a small filter and a really large filter. So maybe you have a filter that's like 6 inches around, and then you have a filter that's much, much larger, right? And you pour that salty sand water over these two filters. Which one is going to filter that water more quickly? I think it's pretty obvious, right? That the larger the surface area, the bigger the filter, the more quickly that filter can do its work. This idea is really important in digestive processes. Let's think about digestion for a moment. In the small intestine, as food travels through the small intestine, food molecules are absorbed into the body and into your bloodstream. Well, the small intestine is not a smooth tube. It's a tube that has a lot of ruffles and bends and pleats in it. And those bends and pleats and indentations are there to increase the surface area. And that allows for even more nutrients to be quickly absorbed into your digestive system. So you'll see a common theme in your body in many places where there's an increase in surface area that allows for increased filtration or absorption or diffusion of molecules. And then lastly, maybe I should have put this one at the top of the list. If we're dealing with a membrane, again, a filter or a cell membrane, if the molecule is too big to go through the filter paper, it doesn't matter how long you sit there and wait, right? The filtration will never happen. So if the membrane is impermeable, then clearly diffusion would never happen. The bigger the openings, the bigger the holes, the more opportunity there is for the molecule to move across the membrane, the greater the rate of diffusion. So those are 5 different phenomena that can affect the rate of diffusion, and they all work together, right? So these would all be occurring simultaneously. Another type of passive transport that does not require energy is osmosis. Now, I know for a fact that you've been learning about the definition of osmosis since somewhere in the middle school years. So this is familiar to you, but perhaps you didn't think about it in the way that I'll be presenting it here today. So osmosis is the movement of water across a membrane. So here we're talking about a membrane. Think cell membrane, right? All the cell membranes in your body, all the cells have a membrane. Osmosis is the movement of that water across a membrane in response to a difference in pressure. Let me help you with this. When you're thinking about osmosis, I only want you thinking about water. Ignore the other molecules that are involved in this picture. Okay, so just think about the water molecules themselves. Ignore the solutes. So ignore the salt or the protein or the other molecules that are also in the environment. Think just about those H2O molecules. When you're thinking about osmosis, I want to make sure these 3 words make sense. So a solute is the stuff that is dissolved. The solvent is what everything is dissolved in. So in your body, we have an aqueous environment, right? Most of our cells are largely water. Most of the molecules in our body are dissolved in water. So in that conversation, all the molecules dissolved, for example, in our cells or dissolved in our blood are the solutes, and they are dissolved in water, right? So water is the solvent. And when you take a solute and you add it to the solvent, you make a solution, right? So a solution of salt water, right? The salt is the solute, the water is the solvent, and together they make a salt water solution. So let's think about this osmosis a little bit. And if I were to say to you, I had a solution that was composed of 10% protein. Now, I'm making this as simple as possible. But if I said to you, I had a solution and it was 10% protein, you could assume, right, that it was 90% water, right? So if something is 10% protein and it's in water, then it must be about 90% water. And likewise, if something is 85% water, we could assume that it's 15% protein. So just with that simple math in mind. What happens in osmosis is that water will move toward the lower water concentration. In other words, water will move from where water is in higher concentration to where water is in lower concentration. Again, think only about the water molecules. Okay, think only about the water molecules. So water will go from where it is, where water is in higher concentration, to where water is in lower concentration. Now, the phrase that I have always used to describe this and what helps me remember this is that solutes suck. That is, the higher the concentration of solutes, the higher the pressure, and that is going to pull the water toward it. So if you have a high concentration of salt, the water is going to be drawn toward the salt, toward the solutes. The solutes are going to suck the water toward the higher concentration of solutes. So let's look at this in a diagram. We're back to a beaker. The beaker has, as you can see, a dividing line. Okay, there's a membrane here of some sort. But what's important here is to realize that this membrane is not permeable to the orange solute molecules. This membrane is permeable to water. So these small little bluish spheres are water. And then these orange spheres would be some sort of solute. What's important again is that this membrane is not permeable to the solute, but it is permeable to water. So, if at time 0, you put a lot of solute over here on the A side, the B side you put nothing in there, what's going to happen? Well, water from the B side is going to be drawn over to where the solutes are. Like I just said, solutes are going to suck water from the right side over to the left side. Another way of thinking about this is that water is going to go from where water is in a higher concentration. Do you agree that over here on the B side, water is 100%, right? There's 100% water over here. There's nothing else. So water is 100% on the B side, and over here, let's just put a number up here, it's 85% water because there's 15% of those solute molecules. So what's water going to do? Water is going to go from where water is in higher concentration to where water is in lower concentration. So it doesn't matter which way you're thinking about it. Think about it. Water is going to go from higher concentration of water to lower concentration of water, or you think solutes suck. You'll always get the right direction of movement when asked about these sorts of ideas. So if this membrane is not permeable to the solute molecules, but only permeable to the water, then what would happen? There would actually be a volume change here. You would actually see water volume go down and water volume go up on this side. Okay. Now, in your body, this is the basis of edema, of swelling, right? Water moving from one area to another area, to an area where actually there's more fluid than you want, right? So if there is a swelling, it's because there's an influx of water, and there's a biological reason for that, and we'll come back to those ideas when we get to the lymphatic system. But I want you to see that this isn't just about salts in a beaker, but there are actual physiological human biology phenomena that are they're going to follow these rules of the movement of molecules across the semi-permeable membrane. Okay, so that is a little bit about diffusion and a little bit about osmosis. Here's another concept about how molecules can move, or how water can move from one area to another area, and this is referred to as tonicity. Tonicity is defined as the ability of a solution to affect the volume and pressure. And for this conversation, I want you thinking about cells, and I'm going to show you some cells. Okay. So In this simple diagram, I have a cell. The cell is indicated by this little orange hexagon. So there's a cell. The cell is sitting in an environment. So this beaker or this rectangle represents an environment for that cell. And what I'm telling you is that the solution here has a lower concentration of solutes than the intracellular fluid. So intra means within. So within this cell, okay, the solution around the cell has a lower concentration of solutes than the intracellular fluid. So what I'm going to say here is that this is pure water. Let's say this is 100% water out here. Okay, 100% water. And cells are not 100% water, so let's just say for the purposes of this, the cell is 70% water. Okay? And then it has 30% of other solutes. Salts, sugars, proteins, nucleic acids, all the things that cells have in them. What's going to happen here? Well, based upon what we just described, water is going to go from where water is 100%, right, where water is in higher concentration, and water is going to move into this cell where water is in lower concentration. Or, the other way of thinking about it, and we get the same answer, is that inside this cell there are solutes, and solutes are going to suck water into that cell. So what's going to happen to that cell? That cell is going to swell. It is going to get larger. It is going to increase in its volume. And so what we're going to say is that cells that are placed in a hypotonic, so remember hypo means lower or less. Tonicity, so the water has less stuff in it. It is pure water. There's less stuff. It's 100% water. There's zero stuff. We would say that's a hypotonic environment. And the cells there are going to, they're going to increase. Okay, they're going to enlarge. And if the influx of water is so rapid and there's such a difference in concentration that so much water goes in, that enlargement can lead to a lysis. That cell can lyse, break open, pop. The way I remember this, guys, is in a hypotonic solution, the cell becomes hippo. I know it's really weird, but the cell enlarges in its size. Okay, it enlarges. And here is a red blood cell. Now, I've got this little backwards. You'll see in a moment what a normal red blood cell looks like. But what you're going to notice as we go through the next couple of slides is that this red blood cell is perfectly round. It looks like a big old tomato, right? A big round kickball. And your red blood cells do not normally look that enlarged. So that cell has definitely taken on fluid because it was in a hypotonic environment. Let's look at the opposing situation, a hypertonic environment. So hyper, more. So if our environment, before we said that our cell was 70% water. Okay. Let's say now that out here we have only 50% water in the environment. So we have 50% water and 50% solute. So this is a very salty, very a solution with a lot of tonicity, a lot of concentrated solutes in it. And the cell, just like our cell before, has 70% water, okay, and 30% solute. Okay, so based upon what we've talked about, what's going to happen to this cell? Well, remember, there's two ways to think about this. One, water is going to go where it's in higher concentration and where water is in lower concentration. So that means that water is going to move out of the cell, doesn't it? Water is going to move out of the cell. The other way of thinking about it is that solutes suck. And because there is a greater concentration of solutes out in the environment, again, water is going to be leaving the cell and coming out into the environment. The cell's going to shrink. Okay, so cells placed in a hypertonic environment will shrink. Now the word for that is the cell is going to crenate. Okay, the cell's going to crenate, or it's going to undergo crenation, right? And when a cell crenates, it becomes smaller. And the cells I'm showing you are red blood cells. And red blood cells are doing this all the time. They are very flexible cells. As red blood cells travel through your blood, they're flexing and changing their shape a little bit as they travel through your bloodstream. And red blood cells are also very sensitive to changes in tonicity. So if a red blood cell is put into a hypertonic environment, into a salty solution, then what happens? The cell crenates. And you can see these spiky, the spiky appearance of this cell. This is not a normal looking red blood cell. You can see the spikiness to this cell. The third example is isotonic. Well, maybe you know that iso means same. So when you have a cell that is 70% water and you put it into a solution that is also 70% water, they're equal, right? Isotonic. And at this point, yeah, just like in that equilibrium or that diffusion example I showed you earlier, there might be a few molecules moving out, but there'll be just as many molecules moving into the cell. And so nothing's going to change. The cell is going to stay the same. And when you look at that cell, it is going to maintain its normal shape. So here is the normal appearance of a red blood cell. You'll see a lot more about red blood cells in the course. But there's actually an indentation in the cell. It kind of looks like a river tube. A red blood cell from the side is said to be biconcave. Kind of looks like a dog bone. And it has indentations on both the bottom side and the top side. In that crenated cell, that red blood cell gets these spiky appearances, right, and gets smaller. And in the hypotonic environment, that cell swelled up to become a large sphere. So that is the idea of tonicity. Tonicity. And this will become important when you are, if you're going into nursing or into healthcare and you're thinking about IVs, right? What sort of IV are you putting into the person's vein? And what is the concentration of solutes? And what effect could that have on the red blood cells as they interact with that IV solution? If you were to put a very salty solution into someone's veins, it would actually cause their red blood cells to pop, to lyse. And if you put a I think I just said that wrong. If you gave someone a pure water, right? The reason we don't give pure water in our IVs, I apologize. If we were to put pure water into veins, then that's a hypotonic environment and your red blood cells could potentially pop. Conversely, if you were to give someone a very salty, high salty solution into their veins, that'd be a hypertonic situation and you would expect the red blood cells to crenate, to shrink. Also in our biology basics presentation, besides diffusion, osmosis, and tonicity, I want us to think about solubility. That is, we know that oil and water don't mix. Right? You're learning in lecture about polar and non-polar molecules. So water is a polar molecule. We know what that means. We know that if we think about the Mickey Mouse sort of appearance of water, that the oxygen is slightly negative in charge and the hydrogens are slightly positive, right? The two hydrogens are slightly positive. So this positive and negative difference causes a polarity in the molecule. There's a slight negative charge on the oxygens and a slight positive charge on the hydrogens. This gives the molecule an overall charge. There's some negative areas and some positive areas. This is the basis of hydrogen bonds that you heard about. So these dotted lines represent weak attractions between water molecules. And so water is considered a polar molecule, and it has this interaction between the molecules. Oils, lipids, one of the general characteristics of oils and lipids and fats is that they are non-polar molecules. And what that means is that those molecules are evenly sharing their electrons, that there is no overall charge to the molecules. And we all know that oil and water don't mix. And so like dissolves like. Molecules that dissolve in water would also be polar. Molecules that dissolve in oil would also be non-polar. So let's just say this again. We know that in our blood, right, in the plasma, in the liquid portion of the blood, there's a lot of water. Your blood is roughly 92%, the plasma, the liquid portion, is about 92% water. And in that water, you have all sorts of things dissolved, right, or that are traveling through your bloodstream. Here's the basic rule. Like I said, like dissolves like. So water, the molecules that dissolve in water must also be polar. Okay? So here's that hydrogen bond story again. So anything that dissolves in water is dissolving because the molecules are also polar, and they are attracted to and are interacting with the water molecules. So alcohols, sugar, vitamin C, these are all molecules, relatively small molecules, that can be dissolved in water because the molecules themselves are polar. Here's a chemical model example, but here are some water molecules. And then here is this is sucrose, right? There's a sugar molecule. And we know sugar dissolves in water, and it dissolves because the water molecules surround that sucrose and now allow it to be transported or, quote, dissolved in the water. Another way of thinking about it, I like the way that this cartoon shows this, and that is, here is, let's say this is sodium chloride. We talked about this in lecture as well. So we've got this molecule shown in purple here, and this is the solute in purple, and the blue, let's say this is water, the solvent. And we know that a salt crystal will dissociate, will break down into its individual ions, and that will be, each of those ionic components would be surrounded by the water molecules. Now this same situation would happen if this were a non-polar liquid like oil and a non-polar molecule. You would see the same idea of dissolving. The basic way I think about this, if you are painting and you are using indoor water-based paint, and you get that paint on your clothing or you get that paint on your hands, you go to the sink, you turn on the warm water, and you can easily wash your hands. So that water-based paint is water-soluble, water's polar, and that paint is also polar, and that's why they dissolve and you can wash your hands. But if you are painting with oil-based paint or external paint, that is not going to be water soluble. So if you get that paint on your clothing or you get that paint on your hands, you can't go to the sink and wash that oil-based latex paint off. You must use a paint thinner or a acetone or some sort of non-polar solvent to wash that paint off your hands. And that may be the best example that we've all experienced. Okay, a couple more biology basic ideas to go through. Here is the pH scale, not new to you. You've seen this in lecture or you will be seeing it soon. I just want to remind you of a couple things here. Some of the lab activities this week will involve your understanding of the pH scale. Recall this is a scale that goes from 0 to 14. Things that are less than 7 are said to be acidic. Molecule or substances that have a pH greater than 7 are said to be basic or alkaline. Pure water, distilled water, is neutral. It has a pH of 7. Notice that as things become more acidic, the pH number goes down toward 0. And look how this diagram shows that as the pH number goes down, the amount of hydrogen increases. Okay, so you see the arrow getting larger here. So as the pH goes lower than 7, what's happening is that you are releasing more hydrogen. Now, conversely, as the pH goes above 7 toward 14, you see an increase in hydroxyl ion, OH minus. So to go toward pH 14 is to be more basic or more alkaline. Now, if you've taken a chemistry course, you know that there are a couple of different ways of describing acids and bases. For the time here in anatomy and phys, we're going to just use the idea that acids give off or produce or release more hydrogen ion, H+, and that bases give off more hydroxyl ion. Okay, so that's how we're going to be describing acids and bases, and this understanding will get you through all of the problems or thinking that you will have to do when it comes to pH in this course. So what sorts of things around your household, in your kitchen, are acidic? So the things that we eat are acidic, right? We hear this on the TV, right? About the acids that we eat and drink, and that acid, we know, can break down the enamel of our teeth. So things that we eat and drink, most of them are acidic. And if you were to swash them around in your mouth and let them sit in your mouth, yes, those acidic things over time can break down the enamel on your teeth. Enamel on your teeth is much like cartilage in your joints. Once it is worn down, it does not replace itself. So you want to protect the enamel on your teeth. That's why it's not recommended that you just suck lemon juice, right? Don't suck on lemons, because that is a very acidic juice and over time can break down the enamel on your teeth. So what we see is that wine, sauerkraut, beer are quite acidic. Soda is also very acidic. Things in your refrigerator, cheese, yogurt, milk are also weakly acidic. Again, water is neutral at pH 7. One thing that I want you to know specifically is that blood has a pH of 7.4. That is, blood is slightly alkaline or slightly basic. Blood is not neutral, right? It's slightly alkaline or slightly basic. And then other things around your house that are very basic would be, for example, milk of magnesia. Now, why would one take milk of magnesia? Yeah, you take milk of magnesia if you have an upset stomach. So you have an upset stomach, usually caused by acid in your stomach, and you take some milk of magnesia, and that is going to help to neutralize those acids and hopefully end the stomach discomfort. Oven cleaner is also very, very basic. If you've ever used oven cleaner, you know that it's very slippery. You should be wearing gloves when you put oven cleaner into the oven. So as a general rule, you don't want to be swimming in or playing with strong acids or strong bases. But should you get oven cleaner in your hands, at first it will just feel very slippery. But if you don't wash it off very quickly, it will begin to really erode and affect your skin. Likewise, if you were to swim in very acidic things, you would start feeling your fingers kind of getting tingly and prickly, and literally you're burning your skin as well. So you would want to wash off acids off your hands, like battery acid, something like that. Get that off your hands as well. So that's a little bit about pH. And if we turn this scale from 0 to 14 on its side, again, things that are down here in the low numbers are increasingly acidic. Things that go above 7 are increasingly basic. I've already talked a little bit about pure water being 7, right? Most of the food items we eat are going to be acidic. Many of the chemicals around our house are basic. We have 2 more topics to get through. This one is on the cell cycle. So we're switching from transport, we're switching away from pH, now introducing the idea of cell cycle. So you have trillions of cells in your body. Every one of those cells has its own life cycle, referred to as the cell cycle. And there are 4 phases involved in that cell cycle. There's G1. Some books will refer that as the growth phase. Okay, the growth phase, G1. There's an S phase, S for synthesis phase. There's a G2 phase, which is a second growth phase, or some books will call it a gap phase. This diagram uses the word gap. And then the 4th stage of the cell cycle is a process or a series of events referred to as mitosis. Mitosis. So in this diagram, this is representing the life cycle of a cell. Let's just say this is one of the epithelial cells in your skin. And so the cell cycle begins with G1. So the cell has just been formed, and it's going to grow. It's going to do its normal metabolic processes. And if that cell begins to receive the correct chemical and hormonal signals, the environment's right, and all things are good, then that cell is going to prepare to divide at some point in the future and will enter into the S phase. Now, during the S phase, the DNA is going to be replicated. That is, all of the DNA in the cell is going to be copied into an exact replica or into an exact copy of the DNA. During this S phase, other organelles are also formed. So more ribosomes are formed, more mitochondria would be formed. So you're basically going to be doubling the organelles and doubling the DNA. After that S phase, the cell goes into a second G phase, the second growth phase, and this is where the cell is going to continue to grow and just wait for that mitosis or that mitotic event to occur. Along the way, there are what's called checkpoints. The cell is checking out its nutrient levels, it's checking out the environment, it's making sure that there aren't any, quote, stop signals coming to the cell to not divide. And if all of the green lights are on and the cell is still getting the right signals to divide, it will go into mitosis. And we're going to look at more mitosis a little bit more carefully. And mitosis itself is divided up into some sub-phases. So we'll look at those in a moment. Now, in a rapidly growing cell, like an epithelial cell in your mouth or in your stomach, this entire cell cycle can be 8 hours. Okay, so some of the fastest growing cells in your body can double and duplicate and replicate themselves in about 8 hours. Other cells in your body might take days, months, years. And there are some cells in your body that once they are formed, they never divide again. Okay, so one example of cells that never divide again are neurons. And we'll talk about neurons a lot when we get to the nervous system, but the main cell of your nervous system, the cells that send electrical signals and are in charge of so many things in your brain and spinal cord, once they are formed, they do not once again divide. So we see that the cell cycle can be from hours to years to it never happens. Let's take a look at mitosis more carefully. So mitosis, the word means thread. And what that refers to is that when scientists first looked into a microscope and they were able to observe cells undergoing mitosis, they noticed that the cells had these thread-like substances in them. They didn't know what they were. And from that thready word, we get the word mitosis, something to do with threads. We now know that those threads that one sees under the microscope are chromosomes. And in your cells, in my cells, we have 46 chromosomes, 23 that came from your father's sperm and 23 that came from your mother's egg. And those 46 chromosomes combined to give you a normal cell with a normal number of chromosomes. What you're seeing in this diagram, all these red and blue look like X's, if you will, in this picture. Those are some chromosomes. Now, it's a very simplified picture here. It's not showing 46 chromosomes. It's just showing 4. And we see that the chromosomes are found in the nucleus. So we see a purple-colored nucleus over here. Well, what happens in prophase is that the nucleus is going to break down. So I need you to know what different things are happening in the cell during these different phases. So in prophase, the nucleus is going to, I'm just going to say the word dissolve, right? It's going to break down. And these organelles, the centrioles, are going to, the centromere pairs are going to now head to their respective poles. And the third thing is that the chromosomes are going to become visible under the microscope. Typically, when you look at a cell, you're not able to see any chromosomes. But in mitosis, the chromosomes become visible. They condense. They wind up very tightly, so densely that they're now visible to you under the microscope. So during prophase, we're preparing for division. Then the cell goes into a very characteristic phase called metaphase. And what happens is that the chromosome, now notice the nucleus is gone here, and the chromosomes all line up perfectly along the equator of the cell. So cells, interestingly, are not just a ball that rolls around, but cells have a top and a bottom. They have their, quote, poles, like the Earth has the North and South Pole, and, like the Earth, cells have an equator. And the chromosomes line up along that equator, and the centrioles are now at the two poles, North and South Pole, if you will. And the centrioles, made of microtubules, are going to reach out and grab these chromosomes. Now remember that these chromosomes are already duplicated. Remember back in the cell cycle, we said that in the S phase, that the DNA underwent replication. So that means that when the cell enters into mitosis, it already has duplicated chromosomes. So these are duplicated chromosomes, so that in the next phase, anaphase, as the chromosomes are pulled apart, right? You see that as the cell begins to divide, it starts to pull. And these chromosomes have already been duplicated. So really all that's happening is that the top is pulling one set of chromosomes and the bottom is pulling another set of chromosomes. In anaphase, you begin to see what's called cleavage. So cleavage is simply the indentation of the cell as it begins to break into two. And you can see that the cell becomes more elongated, starts to look more like a rugby ball. Then finally, there is telophase. Now, telo means end or last, so it's the last phase of mitosis. And you can see that the cleavage is much more dramatic in telophase. The two cells are pulling, or that cell is pulling into two cells. And the exact opposite of what happened in prophase happens in telophase. That is, the nucleus is going to reform. So you see the nucleus is reforming here around the chromosomes. The chromosomes, I'm going to call them C-somes here, the chromosomes are going to unravel, or that is, they're going to become invisible again. You won't be able to see them under the light microscope. And the cell is going, the one cell is now going to become two cells. Now, when those two cells are finally formed, when that one cell finally splits into two cells, that marks the moment of cytokinesis. You may recognize the word kinesis, like kinetic movement, like kinesiology, the study of movement. And so cytokinesis is simply the moment when these two cells become two separate cells. Now, the moment that these cells finish mitosis and undergo cytokinesis, then each of these daughter cells would then be said to be in G1, right back to the beginning of its cell cycle. And again, around and around and around we go. Every cell in your body is somewhere in its own cell cycle. So what will be expected of you is to grab your atlas, grab your textbook, grab your lab manual, and take a look at these different stages of mitosis and know the features that's happening in each of these stages. So here, I can see the chromosomes are lined up beautifully in a straight line, right? So this one, that's mitosis. Or sorry, metaphase. That's metaphase. Then here I can see that the chromosomes are being pulled apart. The cell's more elongated, and I can see the chromosomes being pulled apart. That is anaphase. Here I can see the very deep cleavage. The cell appears to almost be forming two cells. The chromosomes are very much separated, and while you can't see it yet, soon the nucleus would be reformed around those chromosomes. That's telophase. And then, if none of those things are happening, if you don't see the chromosomes lined up, if you don't see them being pulled apart, if you don't see them distinctly separated as in telophase, then we're going to just say that that's prophase. So in prophase, right, all the chromosomes are visible in the center, but I don't see any of those other characteristic views, so we'll call those cells prophase. Now, if you look under the microscope at a ball of cells and you see a cell where you can see the nucleus, but you don't see any chromosomes at all, that cell would be an interphase. So if you go back and look at that cell cycle picture, right, let's go back and look at that cell cycle picture. If you were to look under the microscope where any of this green is, you would not see chromosomes. You would see a nucleus, it would be there, but you would not see any chromosomes. And this period where the green is collectively referred to as interphase. Interphase. During interphase, mitosis is not occurring. So another way of thinking about the cell cycle is that it's interphase followed by mitosis. Interphase followed by mitosis. Okay. Remember, interphase would be the collection of G1, S, and G2. So as your cells go through their life, right, they're going through G1, S, G2, then going through mitosis where they form two new daughter cells, which then start their own G1 phase. Okay, so lots of concepts in this lab. The last concept brings us to field of view, and this is a microscopy idea. In your lab materials, there is a field of view worksheet. And then there's another folder that says Field of View Worksheet Answers. So I'm going to describe to you what I want you to draw on the bottom of that first page. So on the bottom of that Field of View worksheet, you see 3 circles like is here on the screen. And go ahead and add these comments to that, either to this PowerPoint slide or to that worksheet page. So these 3 circles represent what you see when you look into the microscope. Right? You look into the microscope, you see a circle of light. That is called the field of view. And here I have the field of view when you're seeing things under 40 times total magnification. Remember that if you're seeing things magnified 40 times, you are using the 4x objective. If you are seeing things magnified 100 times, you're using the 10x objective. And if you are seeing things magnified 400 times, you are using the 40x objective. In this exercise, what I want you to imagine is that you're looking into your microscope, you're using the 4x objective. That means you're seeing things magnified 40 times. And if you were to put a clear ruler onto your stage and focus into that ruler, how many millimeters would you be able to see from edge to edge across this field of view, this circle of light that you can see? And if you were to do that, you would get something really close. You would see just about 5 millimeters across. So the entire circle of light from one edge to the other edge would be 5 millimeters. Now, if you then turned your objective up to the 10x objective, and you still are looking at that ruler on your stage, and you're focusing on that ruler, you would now, using the 10x objective, only be able to see 2 millimeters across. And then that ruler is still there, and you turn now your objective, you turn up to the 40x objective, and you look, and how much of the ruler can you see from edge to edge? And what you're now going to see is only half a millimeter. Okay, very small. So what I want you to appreciate is the actual size of objects, of cells, when you're using the microscope. You need to remember these numbers, okay? And I'm rounding these numbers up to make the math really easy, but when you're using the 4x objective, you are going to see essentially 5 millimeters across. When you're using the 10x objective, you're going to see 2 millimeters from edge to edge. And when you are using the 40x objective, you are only going to be able to see half a millimeter from edge to edge. Now, there is a mathematical relationship here, and let me just describe that to you. Once I describe this to you, there's a number of problems that you'll find in that field of view worksheet. And I want you to work through those problems and then compare your answers to the answers that are posted. The answers don't just list the correct answer, they also annotate the answer. They describe to you how the correct answer was achieved. So it's very important that you try the problems and then look at those solutions. But if you've ever played with Google Earth, right, you're clicking on the app on your tablet or phone and you see the Earth, and then you start to zoom in. And as you zoom in, you're now looking at North America. Then you zoom in over Michigan, and then you zoom in over West Michigan, and then you zoom in over your hometown. And you can zoom in over your neighborhood and even over your neighbor's house, right? We get the idea that you can zoom in closer. What you're seeing on your computer screen or what you're seeing on your phone screen when you're doing that, think of that as the field of view. And you can appreciate that as you magnify, as you zoom in closer and closer, imagine you're changing the objectives of the microscope to a higher power, higher power, higher power. And eventually, when you zoom in over your neighbor's house or over your house and you see your cars in the driveway, what you can see on your screen, right, is a really small area. And then if you zoom back out, you see the neighborhood, and then you see the city, then you see the state. It's the same idea here, that as you move to higher magnification, what you can see on your screen gets proportionately smaller and smaller. The object gets bigger, but the area that you can see from edge to edge, you can see less of your neighborhood or less of the yard or less of the town. So what is the relationship here? So if I am going from a total magnification of 40 up to a magnification of 100, what fold increase in magnification am I experiencing? Let me rephrase that. If I said I was going from 40x up to 80x, do you agree? You would probably say, oh, that's twice as large. Okay, that's the logic I want you thinking about. So if things go from 40x up to 80x, you would say, oh, that object is now going to appear to me to be twice as large. Well, if I'm going from 40x to 100x, things are going to be how much larger? What fold increase in size am I going to expect? And the answer is 2.5. Okay, so when you go from 40x up to 100x, you are going to see things 2.5 times larger. And when you go from 100x to 400x, you are going to see things magnified 4 times. Okay, so things are going to be magnified 4 times larger. Okay, what's that have to do with the area or the field of view? Well, there's an inverse relationship here too. So if I can see 5 millimeters here when using the 4x objective, and I go up to the 10x objective, that means that my area, my edge-to-edge distance, is now going to be decreased or reduced by a factor of 2.5. So 5 divided by 2.5, right? 5 divided by 2.5 is in fact 2. Right? If I divide 5 by 2.5, I get 2. Likewise, when I go from 100x over to 40x, I am going to be reducing my area by a factor of 4. So 2 divided by 4, right? 2 divided by 4 is a half. So there is a mathematical relationship here. Now, if that's not making sense to you right now, that's okay. Go ahead and write this down, scribble this down the best you can. And then let's just take a look at this last slide together. This represents one of the problems that you are going to be asked to figure out here. So let's read this, work through this together, and that will end our conversation today. So the question is asking, it says, if this is the field of view under 40x total magnification. So what it's saying is that this circle on the left represents what you're seeing when you look through the microscope and you're using the 4x objective. Remember, this is 40x total magnification. So you're using the 4x objective. Look back to that slide before, and it says this cell or this object is approximately how many millimeters wide? Well, remember that when you're using the 4x objective, what you can see from edge to edge, what did we say it was? 5 millimeters. Okay, this is very practical. Okay, so approximately how wide is that cell, this circle, this thing that I'm seeing in my field of view? Well, this is very rustic, very rough, but if I just draw some more of these circles, right? Roughly, approximately, right? Approximately, that cell or that thing, do you agree, is about 1 millimeter wide? Because I can draw roughly 5 of those objects across. Now you could argue, and you're not wrong, you could argue that I could draw 6 of those things, right? And if I can get it perfectly or not, but I could argue that I could draw 6 of these things across. And so really that isn't 1 millimeter, that's really more like, 0.8 millimeters. Let me assure you that when I ask you a question like this, I will not have difficult math involved. And if you put anything from about 0.8 millimeters to about 1.2 millimeters, I would accept that as a correct answer because I'm looking for approximate values. But the only way you can answer this question is to remember. You need to remember when you're answering something like this that this is 5 millimeters across, right? That's what you have to remember here. That when you're using 40x total magnification, and that means you're using the 4x objective, that this is 5 millimeters across. Now, let me ask the question in a different way. Let me just change the question. What if instead this did not say 40x, but this said 100x? How would that change my answer? So if I am looking at this field of view and it's saying it's for the 100x, recall that means I'm using the 10x objective. Looking back, that means that this is 2 millimeters across. And if that entire field of view across is 2 millimeters, and I can draw about 5 of these, right? That hasn't changed. I can draw about 5 of these objects across. 2 divided by 5, right, would be 0.4. So that object would be roughly 0.4 millimeters. So that's the extent of the math that I'm expecting you to do. It's not difficult math at all. Some of the problems on the worksheet ask you to convert between millimeters and micrometers. If that is a challenge for you, don't worry about those conversions. That will not be something that I will ask you to do on your own. But I do explain those solutions on that field of view worksheet answer form. Okay, so this has been a long video, I know, but we have a lot of things to cover in this video. Look back in the lab folder, look over the pages that you must complete for this lab, as well as the mastering assignment that's associated with this lab. Make sure you get that done by the deadline. And if you have any questions about this material, your lab instructor will be glad to discuss this with you or your SI leaders. Have a great day.